Welcome everyone. We would like to thank initially for the provision to the Helmholtz facilities as part of the BMBF-funded DPPN-Access 2.0. Within this project, named UV Protect, sunlight simulation of elevated UVB radiation was performed to dissect the metabolic landscape as well as fitness within a global Arabidopsis thaliana population to identify novel UVB protective components as well as understanding the sole impact of UVB radiation on plants. To start with, UVB has a detrimental impact on plant fitness and is harmful for DNA that can result in mutagenicity in exposed tissue. Furthermore, it can cause cell death and apoptosis. In addition, induction of senescence-associated gene as well as reactive oxygen species are accumulating. To emphasize on the global impact of UVB, we highlight in the right panel various information regarding UVB. Coming back to natural products, plant developed an arsenal of chemical compounds that protect themselves against various stresses and environmental stimuli. In a formal study from our lab, we demonstrated that different accessions of Arabidopsis thaliana produce different levels of specialized metabolite as depicted in panel A. What strikes out in this heat map is that a compound class named cygenols behave in a qualitative manner where only a subset is producing these compounds. After purification, the structure was elucidated as phenylacetylated flavonoids as demonstrated in panel B. The gene causing for the production of this compound was named FPT2, flavonol phenylacetyltransferase 2. FPT2 can accept various substrates ranging from chemiferroglycoside to isorhamnetin glycosides and binds a range of different phenols, namely coumarate, caffeate, and synapate, demonstrating enzyme promiscuity. Furthermore, cygenols demonstrate specificity in terms of tissue in their accumulation, in which reproductive organs produce the highest amounts. Looking at the global distribution of the accessions with indicating producing and non-producing lines, it became evident that producers are exposed to higher irradiation of UVB in their natural habitats compared to non-producers. To investigate if indeed geographical adaptation to higher UVB exposure regions led to the emergence of UVB protective compounds, and to further elucidate identification of novel regulators of UVB signaling, we utilized in this UV Protect a global population of Arabidopsis accessions of over 300 lines and performed genome-wide association mapping by integrating UVB exposure as different environments to further dissect the proportion of genotype by environment interactions. Briefly speaking, plants react to different environments in varying degree of trait expression. Different responses on the genotype level to changing environments are called genotype by environment interaction. In our current project, we define environment 1 and environment 2 as UVB exposed and non-exposed environments to dissect its importance. What is also key is that the pattern of identification of quantitative trait loci can vary depending on the environment. These can be either antagonistic, conditional neutral, vary in their sensitivity, or behave similar in a given changing environment. For this, the environmental simulation facilities of Helmholtz Munich provided perfect conditions to dissect the two defined environments of a population scale, as this was demonstrated in the previous study where the sole impact of UVB was dissected while keeping the overall light quality similar to the controlled condition. These experiments were performed in the exposed screen chambers shown here. As shown previously, different accumulation have been observed for different tissue. Therefore, we collected vegetative and reproductive tissue as well as seed to determine the population on various layers. Currently, the experiments have been successfully performed and samples are being processed. The sheer number of biological samples enable us to untangle the sole impact of UVB on plants. This data will be ultimately enabling further the identification of novel UVB protective compounds in plants. With this, we would like to thank the collaborators from the Helmholtz Institute for their immense help in performing this large-scale, labor-intense project, as well as the funding sources from the DPPN-Access call funded by the BMBF. Thanks.